we can start the talk from the definition of sperm DNA fragmentation, which is the presence of single and double-stranded DNA breaks into sperm nuclei. We know that currently many laboratories detect sperm DNA fragmentation as an adjunct to routine semen analysis. The last version of WHO manual for semen analysis added test for SDF detection within the male infertility diagnosis. Indeed, we well know the limitation of routine semen analysis to establish the male fertility status, and these limitations are not surprising because, as shown in this picture, the fecundating potential of an ejaculate depends on many sperm traits besides conventional semen parameters, and several of these traits are acquired after ejaculation into the female genital tracts. And when spermatozoa arrive to the oocyte, they have to deliver an integer DNA and a proper epigenetic cargo, which is crucial for fertilization and next embryo development. So generally speaking, we can say that spermatozoa have to simultaneously accomplish many requirements in order to be able to fertilize the oocyte and to support successfully the next embryo development. Thus, it is unlikely that test just one single parameter is sufficient to evaluate the fecundating potential of an ejaculate. On the other hand, the presence of a relevant sperm defect has to be extended to the entire sperm population in order to impede reproduction. Another important point is which is the sperm population should be tested. We know that only a very small sperm population reaches the oocytes and with competence for fertilization, and in principle, this sperm population should be the most suitable population for diagnostic tests. So, in this complex scenario, it's quite surprising to me that detection of just one parameters, namely sperm DNA fragmentation, in the bulk of native semen samples does predict natural pregnancy. So the question is, why does sperm DNA fragmentation predict natural pregnancy? A possible explanation comes from the tip of iceberg theory. This theory was first proposed by Evenson in the early 2000s, and he proposed this theory to explain why SDF amounts higher than the 30% threshold blunted the probability to get pregnancy, albeit the remaining 70% of spermatozoa with apparently intact DNA. In this theory proposes that the detected SDF is only the visible part of a more standard abnormality. There is a submerged damage somehow impacting reproduction. According to this theory, we can think that only when SDF is equal or higher than a certain threshold, the submerged damage is extended to the entire sperm population, and thus the probability to get pregnancies is very, very low. On the other hand, when SDF amounts are lower of this threshold, there is the possibility that there is a fraction of sperm without defects related to sperm DNA fragmentation. However, the pregnancy is not guaranteed because other types of sperm defects not related to sperm DNA fragmentation can be present and impede reproduction. This theory can also explain why SDF detected in non-viable spermatozoa predicts natural conception similarly to sperm DNA fragmentation detected in viable spermatozoa. And this is a quite surprising result, because non-viable spermatozoa should have the possibility to reach the oocyte and thus to impact reproduction. Again, we can think that the detected non-viable SDF just signals a more extended submerged damage behind the detectable one. The existence of this submerged damage can be revealed when spermatozoa undergo an insult, for instance, during in vitro incubation or during selection, as in the case shown in the slide. In this slide, the graphs shows that density gradient centrifugation increase the damage in some subjects. Of course, all subjects undergo the insult of density gradient centrifugation, but as you can see, only certain subjects increase the damage. And we can think that these subjects are those have this submerged vulnerability or damage, as this scheme shows. In this scheme shows two hypothetical subjects. In the first case, selection delete non-viable DNA fragmented spermatozoa and thus decrease the amount of SDF. In the second case, we have deletion of this spermatozoa as well, but the presence of this submerged vulnerability induces an additional sperm DNA fragmentation amount. The nature of this submerged abnormality is not known. We can hypothesize a poor protamination or maturation. In any case, I think that it deserves a further investigation because it has the potential to affect male fertility. Now, let's see test to reveal SDF, starting from SCSA. This test works only in flow cytometry, requires a previous step of DNA denaturation, and then samples are stained by this stain, acridine orange, which is a very particular stain because it emits green fluorescence when bound to native double-stranded DNA and shifts to red fluorescence when bound to denaturated single-stranded DNA. So for each cell, it is possible to calculate the ratio between denaturated and total DNA, and this ratio is called DFI. And DFI is represented as a frequency histogram, where the percentages of spermatozoa with DNA fragmentation are calculated as a cell outside the main sperm population. This test doesn't detect the real DNA breakage, but rather the susceptibility of sperm chromatid to the induced denaturation. We can skip this slide and pass to tunnel. Tunnel is the labeling of the ends of single and double-stranded DNA breaks with fluorescence nucleotides, and uses both fluorescence microscopy and flow cytometry. But the two types of instrumentation don't give the same results. In particular, flow cytometry is much more sensitive, and again, results are percentages of spermatozoa with DNA fragmentation. Doesn't work. Can you? Okay. Okay, thank you. So let's see alkaline comet assay. This test requires inclusion of cells into agarose, and then a step of lysis, chromatin decondensation, DNA unwind in alkaline conditions, electrophoresis, and DNA staining. During electrophoresis, DNA fragment migrates towards the anode, and thus, if the cell has DNA fragmentation, a pattern like a comet is formed. In this case, the results are percentages of fragmented DNA per cell, so quite different to the other tests, even if some authors use the percentage of comets on total spermatozoa, so come back to a similar results of the other test. SCD test requires the inclusion of cells into agaroses as well, and then a step of acidification and lysis, which remove protein from nuclei, and DNA staining. It uses light or fluorescence microscopy, and after sample processing, spermatozoa with DNA breakage show no halo or small halo, and spermatozoa with integer DNA show bigger halo. And results are again percentages of spermatozoa with DNA fragmentation. Help me. Okay? All these tests are unable to discriminate between single and double-stranded DNA breaks. Double-stranded DNA breaks are believed a more severe damage, and some studies associate this type of damage to an increased risk of miscarriage. And usually this type of damage is detected by the neutral version of comet. However, recently two novel tests have been published. These are similar, they are both a similar modification of a CD test, and they detect only double DNA breaks. In the first one, published in Andrology, spermatozoa with double DNA breaks are detected by a larger halo. In the second one, published in CREP, the spermatozoa with double DNA breaks are detected by the presence of the halo. As we know, a gold standard test to detect sperm DNA fragmentation is not established. And so what we can do is to compare the different tests from different point of view, starting from the type of revealed damage. Generally, it is believed that tunnel and comet assay reveal the real DNA breakage, whereas a CSA and SCD test detect anomalies in sperm chromatin. From the point of view of the reliability, a CSA is the only test relying on a standardized procedure which minimizes the variability among different laboratories, and which provides reliable cutoff value. In principle, for the other tests, each lab should build its own cutoff value to discriminate between fertile and infertile men. Point of view of the affordability. Here the winner is SCD test because it is very easy to execute and needs scarce equipment with respect to the other tests. Point of view of prediction of natural conception or natural pregnancy. According to this study, all tests are able to discriminate between fertile and infertile men, but the alkaline comet and tunnel should be the better test in terms of accuracy. And these two tests seems to be the better test for prediction of a clinical pregnancy in couples treated by IVF and ICSI, at least according to these two meta-analyses published in around 2016. However, the impact of SDF on heart outcomes is weaker than that on natural pregnancy, and the studies continue to be conflicting. However, we will have a talk on this topic later on in the morning, and I just want to catch your attention on a particular aspect, which is sperm selection. Now we are aware that selection can induce sperm DNA fragmentation despite yielding highly motile sperm population. In this graph, in this study, we found that density gradient centrifugation increased the damage in 45 men, patients, who subsequently showed a 50% lower probability to get pregnancy. But there is another aspect, that is that subjects increasing and decreasing the damage start from very similar amounts of SDF in native semen sample. This means that very different SDF amounts in selected spermatozoa correspond to similar value in native semen sample. And this aspect could weaken the impact of SDF on heart outcomes in the study detecting SDF in native semen sample, and they are the most part. Unfortunately, there are very few studies comparing detection in native and selected spermatozoa in terms of prediction of heart outcomes. And results are conflicting and apparently depending on the test used. With a flow cytometric technique able to detect sperm DNA fragmentation in viable and non-viable spermatozoa, we study what happens during selection with density gradient centrifugation or swim up. First of all, as you can see, most part of sperm DNA fragmentation in native semen sample is due to non-viable spermatozoa, and during selection these spermatozoa are deleted, thus decreasing the amount of SDF. What happens to the viable sperm fraction? In some subjects, viable DNA fragmented spermatozoa are deleted as well, but in certain subjects we have an induction of the damage, and so producing an additional amount of sperm DNA fragmentation. So we have an opposite situation in native semen sample and selected spermatozoa. In the former, most SDF is due to non-viable spermatozoa, whereas in selected spermatozoa, SDF, if present, is due to viable spermatozoa. And this is another reason for which it would be important to compare detection in these two types of preparation in terms of prediction of heart outcome. And here you can see that with the swim up, 40% of subjects increases the viable SDF, and this percentage increases to 60% with the GC. Last part is regards the mechanism responsible for SDF. This is an important part because treatment for men with high amounts of SDF depends on these studies. Here you can see the three main mechanisms believed responsible of generation the sperm DNA breaks. Recently, a fourth mechanism has been proposed involving external nucleases and acting in the male genital tracts as a part of an apoptosis-like process aimed to delete defective spermatozoa. In the past, we studied the association between sperm DNA fragmentation and signs of the three main mechanisms, and we found, as you can see, a large overlapping between SDF and both apoptosis and chromatin immaturity. Whereas we didn't find any association between sperm DNA fragmentation and oxidative damage. And this last finding was recently confirmed by this study and by another study from my group, where we used the alternative technique to detect both SDF and oxidative attack. We found an overlapping between oxidative damage and sperm DNA fragmentation when we focused the viable sperm fraction. So there is a role also for oxidative attack, and it is in the viable sperm fraction. We summarized the finding of this study in this model. I'm afraid I have no time to describe. In any case, this model gives a great importance to the testicular processes, I mean apoptosis and effect in sperm maturation. As mentioned, there is also this fourth mechanism acting in the male genital tracts, and it is important because if this mechanism will be confirmed, the meaning of the damage produced in the male genital tract would have a completely different meaning. Oxidative attack, as said, is important. It would act in the male genital tracts, and of course it is the main mechanism acting in inducing sperm DNA fragmentation in vitro condition. Conclusive tips and several parts of this talk can be found in this recently published review. Thank you for attention.